Hey everyone, we are going to look at some level 1 science today, focusing on the 2018 genetics variation paper. We'll be covering the entire examination paper in this particular video. Because the exams are actually quite short, and I'll go through this, should be able to go through this quite quickly. So let's get started. Now, question 1. So we have some Punnett squares, so let's look for this. So the allele for rose comb is dominant to the allele for single comb in chickens. Now they normally give you some fancy diagrams, which serves no purpose other than being a fancy diagram, so we don't need them here, and they have copyright reasons as well. Now, so we have two rose comb chickens produce a single comb offspring. They ask you to explain how it is possible. They want you to define the dominant allele, explain the genotype of the parents of the offspring, and use a Punnett square to explain this. So first thing first, if you think about this, the allele for rose comb is dominant, and so the two rose comb chickens, which are the parents, both of them must have at least one capital R. Okay, and then the allele for the single comb in chicken, which is a recessive allele, which is a small r. So we are saying that these two, so we don't know what that is yet, these two produced a chicken, an offspring that has single comb, which is small r. So the only possibility for this to happen, remember, we after sexual reproduction, the offspring inherits half of the genetic information from each parent. So that means this particular offspring must have got the small r from each parent. That means both of the parents must have a small r, otherwise how would they inherit that in the first place? So if we do the Punnett square, then you can see that it is going to be two heterozygous individuals crossing with two heterozygous crossing with each other, and then there's a 25% chance. So you can see 1 out of 4 you have small r, which is, the phenotype is going to be a single comb offspring, which is what we see here. Okay, so if we define what a dominant allele is, so with genetic, it's more about understanding the wording. You can just look at the schedules, like we're going to scroll down and you can have a look at that. I'll break it down as well, but it's all about the understanding. Just highlighting these keywords, dominant. So you got two rows, just figuring out what their genotype is, and then kind of working backwards to figure out what it is. So don't jump into the bullet points first. You're trying to answer the question, how is it possible? So first thing first, you have to figure out what the genotype is. Okay, so define the dominant allele. Dominant allele is the allele that is always displayed. So yes, both of these parents have the recessive allele, but that is being overshadowed by the dominant allele, so they won't be physically displayed. So the genotypes of the parents of the offspring, so the offspring must be small r, because it has a single comb phenotype. It has a physical characteristic of single comb. So that means the only possibility for that to show up is that it has both small r's or lowercase r's. It has both of the recessive alleles. So how is that possible? That means both parents must have a small r each. Okay, so we figure out they have a small r each, and then they both display the rose comb. So that means they must have the dominant trait, and which overshadows or masks the recessive trait. Okay, and then we use a Punnett square to help them with the explanation, which is what we've done already. Okay, so we can quickly have a look. So dominant alleles are always expressed, whereas recessive alleles are hidden or they're not expressed as long as there is a dominant allele. So the single comb offspring must be small r, because it has a recessive trait phenotype. Now, the key thing with biology is the use of vocabulary. You have to make sure that you use a correct terminology to show understanding. Okay, now both parents have to have a dominant allele because they're the rose combs, and they also have to have the recessive allele, because otherwise how would the offspring get the small r's from? Okay, so they have to give a small r to each offspring. In this way, we can see that in the offsprings. Okay, so this is a pretty sure it's a excellent question. Yes, So yeah, so this is a merit question on its own, but if you can do the next part, that go up to the next sense as well. Okay, next question. Explain how a breeder could use crosses to find out if a rose-comb chicken has a pure breeding genotype for the trait. Okay, so first thing first, again, this is a rose-comb chicken, so it has to have a big R, and we're trying to see if it's a pure bred individual. That means it has to be, it's asking you, is it small, big R, because so this is purebred. Purebred, it means that there is no hidden genetic information. Purebred, it means there's no hidden genetic information. Okay, because physically, if you look at the heterozygous versus the homozygous dominant, you can't tell them apart because they display the same physical trait. So how do we, if you want to spend thousands of dollars trying to purchase a purebred dog, if you spend tens of thousands of dollars or hundreds of thousands of dollars on a purebred horse, you want to make sure that your purchase is worthwhile. So how do you ensure that? They're going to give you a certificate, they're going to give you the certificate of the parents, because they're like, yep, my particular animal is purebred, this is how we can prove it. Okay, so pure breeding means there's no hidden genetic information, in this case big R. And the genotype is what alleles, genotype is the combination of alleles. Okay, so combination of alleles, that becomes your genotypes, and then the particular genotypes codes for the physical characteristic, which is known as the phenotypes. So pheno for physical, geno for the genes. Okay, use your Punnett square to help explain when the breeder could be confident of the chicken. Okay, so there are two possibilities. When you do pure breeding, please remember, whenever you see the word pure breeding and then you're trying to find out if something is purebred, you want to cross this with a homozygous recessive. Okay, so must be crossed with a homozygous recessive, because unless you know for sure that a particular individual is heterozygous, that's okay as well. But how would you know? They look the same. So but then if you look at these two, if you have a rose-comb chicken, it's either homozygous dominant, big R, or heterozygous, big R, small r. It has to be one of those two. But if you have a single-comb chicken, it has to be small r, because that's a recessive allele. So straight away you can just look at it and go, yep, that's a recessive chicken, I can breed with that, and then we can do a test cross. Okay, so you must cross with a homozygous recessive, and because they will look physically different, and you can tell straight away, because there are two possibilities. So if you're trying to figure out if this particular chicken is purebred or not, the only two possibilities, it has to be big R or big R, small r. So, and then you can show by the probability, because we are crossing it with only small r's. If your individual was a purebred organism, can you see, using the Punnett square, this is 100%. This is 100% heterozygous, which means there's 100% rose-comb, because you always have the big R. But then if you crossed it with the if you cross a homozygous recessive with a heterozygous individual, can you see you're going to get 50% of small r, which is going to be single comb. Now that means if you do pure breeding, as if any of the offspring, so they can have some offsprings, if any of the offspring is single comb, that means your individual is not purebred, it's heterozygous, because you got the hidden gene combining with the recessive allele, which gives you the homozygous recessive individual. Now if you have, bred 5 offsprings, none of them are single comb, then you can be a little bit confident that possibly you have got homozygous dominant parents, but you can't be sure. The only way to make sure is that you can have many offsprings. Okay, increase the population sample. More offsprings you have, then look at the offsprings. If they turn out to be all rose-comb, they're all displaying the dominant trait, then the possibility of that being a purebred organism is increasing. Okay, but it doesn't guarantee. I mean, you can just due to probability, it doesn't have to happen. Okay, so when they say pure breeding, it means you have homozygous dominant, in this case bigger, just no hidden genetic information. Genotype is the alleles combined together. So the rose-comb chicken must be either bigger, or bigger, smaller. To find if it has a recessive allele, cross it with a smaller combination, because you have 100% rose-comb or 50% rose-comb, depends on the phenotype, depends on the genotype. If any of the offspring is single comb, that means it must be bigger, smaller. If all the many, see the word highlighted, many, because the thing I always tell my students, just say you need to increase the sample size. So once you have a large number of offsprings and they are still all rose-comb, then it's very likely to be purebred. Okay, so that's the end of question 1. Let's move on to question 2. Question 2, this is looking at herbicides. Herbicides are chemicals that are used to kill weeds or many of ryegrass. Explain how variation they develop a resistance to some herbicides so it doesn't kill. Look at that, it's no longer killed by them. Oh my god, they really break it down for you, don't they? The population can help the population develop herbicide resistance. So this is pretty much just a rephrasing, just repackaging the same sort of question that you always say. What is the benefit of having variation? Because you have these variation of the ryegrass, so some of them could have mutated, or some of them could have become more resistant to the herbicide. So you may have a large population, like say for example, you have grass, and then maybe out of all the grass, out of all the individuals, then you have this one is going to be resistant to the herbicide. And then if you're a gardener, then you apply some in this case herbicides. You're trying to kill off the ryegrass, and then what's going to happen is that these guys that are not resistant, they're going to die off. Okay, but then this particular individual that developed some resistant will be able to survive, and then they can pass on their good genes in terms of the resistance. Then if you give that over a period of time, then all of the grass will look like that. Okay, so it's pretty much just saying that this is a benefit of sexual reproduction, because you have some individuals that may develop genes or conditions or phenotypes that may help them survive better, and then they because they survive longer, they can live longer. That means they can reproduce, and then they pass on the genes, and then the next generation will inherit the good genes, and over time they will, the population of the ryegrass in this case will become more and more resistant towards herbicide. And so because if there's no variation, they will just all die. Okay, so that's pretty much a very straightforward question, I hope. So let's have a look. So variation is difference between species, within the species, between population of the same species. In this case, some more resistant to herbicides. So if they're better suited to the condition, in this case, they'll survive better. They pass on the genes, live longer, pass on the genes, the next generation live longer, overall more plants will become resistant, and then there's an increase in plant numbers. If they're all the same, then they all die. That's just the same sort of question every single year. All right, next one. Explain how sexual reproduction increases variation in the population. You should include gamete formation and fertilization. This is actually a really important point. Now, one thing I always do when I was a student and preparing for exams, I always try to think about, if I have to, if they ask me a certain type of question, can I reword it to a different type of question, and then see if I can answer. This is actually a really good question. They ask you to include gamete formation, fertilization, because this are so sexual reproduction produces variation through gamete formation and fertilization. The other type of variation is caused, now I'm not answering this particular question obviously, is mutation. So these two things are fundamentally important to variation. One is through mutation, which is random. One is through sexual reproduction, which is through meiosis and fertilization. Okay, so what is sexual reproduction? Sexual reproduction is combining DNA from two individuals, and then you have to use gametes. So in this case, you have to do meiosis. So the individuals have to make they have to make the gametes. Now the gametes are all unique. Okay, so the gametes are all unique. You can say they have half the number of chromosomes. Actually, let's just write it in. How they're unique. They have half the number of chromosomes of a normal body cell, half the number of chromosomes, and these chromosomes are, each one is unique because of independent assortment. Independent assortment, which is randomly sorting them into assortment, and crossing over, which is just swapping information as they line up, getting pulled apart. So meiosis, you make gametes, and all of these gametes are unique, and then these gametes are going to go into the one lucky one that's going to be undergo fertilization with the other gamete. So you have half the number of chromosomes from in each gamete, and then they combine with another one to make up the full set of genetic information that gives a unique individual. Okay, so the gametes are random. The gametes are all unique because of random assortment, because of crossing over, and then which of the gametes are then used for fertilization is completely random, and therefore you result in unique. That's why you increase in variation. Okay, so let's have a look at the answers here. Sexual reproduction produces combining DNA from two different parents using gametes. These are formed during meiosis. So the parents have two sets of chromosomes, blah, blah, blah. The gametes produced in sexual reproduction have only one set of chromosomes. So what that means, just have half the number of chromosomes. So these can be combined with another one to make a unique individual. This is called fertilization. So when the gametes are formed, so this is where you can go into a bit of meiosis. The answers are really brief. You can talk about the formation, independent assortment. Chromosomes are randomly sorted and they swap information. Each gametes are different, are very, very different. They're all unique, and then which one gets to fertilize is completely random. Hence, you make unique individuals, which results in variations because you have different combinations of the genes. Okay, so hopefully that makes sense. Let's move on to question 3. It's a very wordy examination. I do feel I'm sorry for you guys. If you did the 2018 exam, I don't think there are any family tree, none of that pedigree chart questions. Those are my favorite. All right, so let's have a look at this one. So we're looking at gene mutation. How could a change in gene result in phenotype within the white tui? And then you should include the term DNA and allele. So you need to explain what those words are. So just remember that a gene is a section of a DNA that codes for one characteristic, whereas your DNA is the combination. So your DNA contains many genes. The DNA is essentially the blueprint of life. It contains all of the characteristics, what they should look like, and the gene is a section of a DNA that only codes for one characteristic. Now that's not necessarily true, especially if you go into level 2, level 3 biology, but as a year 11, that's good enough for now. One gene codes for one characteristic. So if you think about the twisted ladder, one small section, that's one gene, and then you have lots of genes in that particular one DNA. Okay, now what is phenotype? Phenotype is the physical structure, it's a physical appearance that you display as a result of the combination of the genotype, or that's what your DNA tells you. Phenotype is a physical structure being displayed, or is a physical characteristic being displayed because of the genes, or because of the DNA, or because of your genotype, whichever words you want to use. Okay, now how does mutation work? I'm just going to give you an example. Like, you remember this ATCG stuff, yeah? I'm just going to randomly give you a combination. Let's say this is some base pairing, and let's just say this is one gene, for example. This is one gene. Yeah. This is one gene. Okay, and mutation changes the base pairing. So it changes the, let's say, after mutation, instead of ATCG, what has occurred is that this one got changed into C and G, and then the other remains the same, for example. So mutation occurred, and then you could have swapped some, you could have deleted some bases, you could have added some bases, some of them got flipped around, lots of different things can happen. But a mutation is a change in the base sequence. Okay, you're changing the base sequence. If you're changing the base sequence, you are changing the protein. If you're changing the protein, you are changing you're changing the phenotype, because the results in the change of the genetic information results in a different genotype, and that results in a different, most of the time you make a new allele, and that will result in a different or new phenotype. In this case, a Y2E, albinism or whatever you want to call it. Okay, so let's just quickly have a look at the answer. A gene is a section of DNA that codes for one characteristic. Phenotypes is the appearance of the gene, or genotype DNA information. Now, a mutation can cause a change in the DNA base sequence for a gene. Now, this results in, normally you create a new allele, because you have different base pairing, you have different base sequence, you result in a different protein, and that results in the change of the phenotype. And one thing that you do need to refer to in this type of question, relate back to the example that you've been given. So you need to talk about the the Y2 would have different base sequence because you have a mutation has occurred, and then the bird has got a different base sequence, so the genotype has changed, which results in a different protein being produced, which results in a different phenotype being displayed, which is why we see a different color. Okay, last question. It's very, very wordy. All right, explain whether the white coloration will be inheritable or not. You should talk about the terms inheritable and non-inheritable. So inheritable just means that something can be passed on to the offspring, and non-inheritable is just it's not in the gametes. So inheritable, this just means the information occur. So the gametes has been affected. Okay, the gametes have been infected. So because of, in this case, whether this could be inheritable or not, I mean, if we go back to the previous question, the DNA has changed, hasn't it? So that means the white 2E has probably got some new alleles. Okay, so it is very likely that this new alleles can be passed on. If this is this information, this change, if this change is passed on in the gametes. So we don't know if it's inheritable or not, because I remember reading this question when the exam just came out. I was like, what? This is rather ambiguous. But I mean, if you think about it, if they say there's a mutation, yep, they just want you to understand if it's inheritable, that means something happening in the gametes. And then in this case, because the 2E has new alleles, because the DNA mutation, we can if that change also occurs in the gametes formation, then it will be passed on. If not, then it won't be passed on. Non-inheritable. So this is I often use this example. Like say, if I have a really loud Year 11 class, like say not me, I don't want to use myself in the example. Like say, if a teacher has a really, really loud Year 11 class, and then this poor sod becomes deaf. Because his class or her class is too loud, is that an inheritable mutation? Like, does that change? Is that change inheritable to the children of that teacher? Obviously not, because it's a physical change. It's just due to the environment, just for that person. Not inheritable, because that's got nothing to do with the gametes. Okay, so if we quickly have a look at the answers. So inheritable means that the information must be passed on to the offspring, and this must occur in the gametes. So white information of the 2E, if there's a change in the genes, because we just discussed it previously, it may be inheritable. Depends if it goes in the gametes or not. Okay. So very wordy examination, but it does test all of the fundamental concepts that you do need to know for this particular standard. The only thing that is missing, which is the pedigree chart, which is a little bit annoying because I do love those type of questions, but it does cover everything else. And I think is, if you get the grip of the basics here, you should be able to get the 4 credits, and surely you can push yourself for something a little bit higher if you just memorize. Biology is all about the terminology, all about the definitions. You need to use the right words at the right time, and then be very thorough with your explanation. It doesn't mean waffle and rewrite the same sentence 20 times. That's not going to help you. So just be precise and memorize the concepts, and you should be fine. Okay, so hopefully this video has been helpful to you. Subscribe if you haven't, and leave a like, and I'll see you guys next time. Bye bye.